Hi, my name is Brayden Guth, and I did my research on clone number 1808, and this clone coded for a gene that had an enzyme important in responding to stress, protein degradation, and senescence. And this enzyme is called an aspartic peptidase. Again, it has three functions, but the functions are more definitive in animals, bacteria, and viruses than they are in plants. But we can still look in a general sense at their function. But to first, I'd like to look at the structure so we know a little bit more about their functions later on. They can be grouped into 14 different groups based on their amino acid sequence homology, and they're composed of either one or two polypeptide chains, but each of these polypeptide chains has two domains in it, and each domain contains a key aspartyl residue, which is just simply an aspartic acid functional group. It is this functional group that is hypothesized to be important in their function because, as we know, things with acidic properties, like that aspartic acid residue, can denature proteins, which is part of the function of these enzymes. Firstly, plants must respond to stress, and they would do so just as humans would. They must also defend and protect themselves too. There are several examples of this, but two specific ones would be pathogen invasion and drought conditions. We know that aspartic peptidases break down proteins, right? So when a pathogen injects proteins into the cell, these enzymes are great at breaking them down, which would kill the pathogen. During drought conditions, these enzymes play an important role in nitrogen remobilization. That is simply moving nitrogen from one part of the plant to another to concentrate it more and keep the plant alive. However, sometimes plants get so stressed that their cells die, and we call this senescence. Several plants have been studied to determine how these enzymes work in cell death. And four of these methods are degrading the chloroplast, triggering cell death, converting proteins to nutrients for embryo and endosperm development, and hydrolyzing other important cell components. This is where things begin to get interesting. Aspartic peptidases are necessary for the breakdown of rubisco. And rubisco is a very important source of nitrogen in plants, more specifically, new organ growth. And as we know from class, nitrogen is one of the essential elements for plants. And this provides a strong argument for the foundation of my hypothesis of why I found this gene in the pollen of male ragweed plants. I hypothesized that since aspartic peptidases are necessary for the breakdown of rubisco, they must also be necessary for cell organ growth. But when would a cell need to grow? And the answer to that is after fertilization. We have a seed, and it needs to grow into a new healthy plant. That is why aspartic peptidases are found in pollen, so then they will be embedded into the embryo and help it grow. But just because these enzymes may be found in pollen is not conclusive evidence that they are allergens. After all, they are found in the cell membranes of all cells and all tissues. And the final thing I would like to point out is that my gene was similar to only 9.23% of the clones found in our cDNA library. Thank you.